guest, Dr. Daniel Getz, co-founder and CEO of Myeloid Therapeutics. The format for this session is a presentation followed by some Q&A. Daniel, thank you for joining us today. Please go ahead.
Yeah, thank you. All right, there's no company presentation up here. Not a good start. Do you have the presentation? It was uploaded earlier today. Oh, there it is. I see it. Yeah, I got it. It just wasn't up. It's requiring a bit more. Well, thank you everyone for getting out of bed early enough to hear what we're doing at Myeloid. We started the company back in 2019, really with the view to take advantage of the innate immune system. Can you see that? Oh yeah, we're good. We're on presenter view. I can see it. Yeah. It's just not here. All right. Excellent. All right, we'll just push on here. All right, fantastic. Yeah, so, okay. We just have one problem. The screen here moves ahead, but not that one. All right, there we go. We're on track. All right. Thank you for getting up and jumping in here this morning. I'm really excited to be here and present what we're doing at Myeloid Therapeutics. It really is, I believe, the next wave of immunotherapeutics. We have seen companies like Moderna, BioNTech, and the likes trying to figure out ways to harness the innate immune system to support CAR T-cell therapy, for example. They're trying to figure out ways to harness the innate immune system to activate T-cells in melanoma, PDAC, so on and so forth, and they've had a lot of early success doing that. And what we're doing is trying to take it to a whole nother level. You know, what I call is in vivo vaccination. The idea that you can arm the innate immune system to see tumors, to liberate neoantigens, and essentially do all of what these other companies are doing ex vivo, in vivo. And what we're doing is really based off an amazing amount of novel RNA and immunological science, which I'll touch base on. We're actually clinical stage. We are the first company to deliver a CAR using RNA directly in vivo, and I'll show you how we're doing that. We have manufacturing solutions using non-dilutive capital to support our programs well into the future. And then the team, really great team. To put into context how amazing the team is, we've put 2 novel platform technologies into humans in the last 4 years, all developed in-house. From the time we did our first animal experiment to the time patient was first dosed with our in vivo CAR was 19 months. I'll skip over this just for time. The real, important element of our technology is harnessing the orchestrator of the immune response, which is the myeloid cell compartment. When you take immunology classes, the very first thing, or one of the very first things you learn is antigen-presenting cells are what tell T cells what to do. And the way these cells function is they have a plethora of pattern recognition receptors. I don't know if this will work. No, it's all good. But you can see within the cell, there's a number of different pattern recognition receptors. You've probably heard of these: TLR, STING, NLRP3. You've heard of companies trying to activate these pathways, usually in a singular or myopic approach, for example, targeting TLR using antibodies bound to TLR agonists. What we have uncovered and what immunologists have known for some time is just like T cells, the innate immune response needs multiple signals to get moving. To get that snowball rolling, to trigger a downstream immune response, you need multiple of these pathways activated. So that's the first thing. The second thing is myeloid cells, whether they are found in the COVID, inflamed lung or in a tumor, all derived from the same blood-derived monocyte. So what does that mean? It means that you have in tumors especially an army of myeloid cells that actually have the capability of being very inflammatory. And what's really interesting is very different to T cells. T cells either struggle to get into solid tumors, or if they do get in there, they struggle to survive and they become inactivated. This is not the case for myeloid cells. In glioblastoma, as an example, up to 75% of the tumor mass can be blood-derived monocytes. Ovarian cancer is about 49%, triple-negative breast cancer is about 45%. So you essentially have a sleeping army of cells that if you know how to activate them, you can actually really harness their ability to be inflammatory. You can remodel the TME. You can actually get them to present antigen T cells. And really, this is what BioNTech and Moderna are trying to do by giving vaccination in combination with CAR T cells as an example. And so how do we go about, trying to solve the solid tumor problem and activate this sleeping army of cells within tumors? There's actually 2 major pillars to our company. The first is the RNA science. When we started the company back in 2019, as you can probably imagine, we were trying to engineer myeloid cells, so we started going down the virus path. It's probably not surprising that these cells are designed not to be infected with virus, and if they do get infected with virus, most of them will turn into macrophages or die. And so we quickly learned that RNA was the way to go, and that's was serendipitous at the time. It was before COVID, and so it was actually a headwind that we encountered early on in the company's genesis using RNA. But the good news for us today is that we have proprietary, receptors, we have RNA optimizations, and I'll touch base on those in a moment. The other element to it is the novel CARs. So all of those different pattern recognition receptor pathways, we've spent a lot of time deconvoluting them and finding, the 15 to 16 amino acid sequence that's really important for triggering the signal within the cell. So you can actually, using RNA, daisy chain a lot of these, and then by obviously jumping on the great work done in the CAR T-cell world, we've built these chimeric antigen receptors that use scFvs to recognize tumors and then activate these innate pathways. And then obviously, if you have RNA and CARs, you can think about the product modalities. And we have in vivo programming technologies and an ex vivo cell therapy that I'll touch on in a moment. When we take all of this technology, what we've actually built out is the pipeline here. And as you can see, we actually have a program targeting TROP2 that's in the clinic dosing patients today. This is an in vivo delivered CAR, RNA LNP construct. We're rapidly moving towards bringing GPC3 and CD70 targeting to the clinic as well, and partnering on EGFR. Our cell therapy program, which I'm going to show in a minute, is something that we're very excited by. It's a very strong proof of concept to show that you can actually program these cells and actually get them to change the tumor microenvironment, and we're moving to partner that with another company as well. So first and foremost, when we started the company, we were really interested in taking myeloid cells and engineering them and then delivering them back into patients because we thought this was an interesting way not only to get proof of concept data, but then also to solve some of the problems within the space today. And when we started, we focused on really one thing, a one-day manufacturing process. So we have actually successfully made a one-day manufacturing process. Cost of goods would be about $22,000, and the vein-to-vein time is 8 days. And we've done this successfully. We've actually dosed 8 patients. And what we found is this is a T-cell lymphoma situation. We found that patients that have been treated with this have more than tripled the mean overall survival now. But what's really most important for us as we're moving forward into doing this directly in vivo was the shift within the tumor microenvironment. So if you can look at the top left here, top right left panel, this is a patient that had T-cell lymphoma. This is a bone marrow biopsy. You can see the bone marrow, the green is all the T-cell lymphoma cells, the blue is your activated myeloid cells, and the red are what we call tumor-associated macrophages. 17 days after treatment is the right. Significant reduction in the green, significant increase in activated myeloid cells, a reduction in the tumor-associated macrophages. If we look at the periphery of T cells, what we found from baseline to day 21 in this case, if you see the orange on the left of the diagonal line, these are TCRs that were not present in the circulation prior to treatment. So this is actually, in our opinion, very compelling information. We need to collect more data, and we're doing that in a partnership basis, mostly because we're very excited by our in vivo technology, and as a company, we can't do everything. We're a small company of 42 people. And so what we've done is we've said, look, how do we take technology and do this directly in vivo? And I've actually been fortunate enough in a previous life to develop nanotherapeutics in the context of autoimmune disease. And during that 10-year experience, we spent a lot of time trying to figure out how do you deliver nanoparticles specifically to different immune cells. And what's quite interesting is the myeloid cell compartment, the spleen and the liver are phenomenal scavengers. It really doesn't matter if you put a CD3 antibody, for example, on the surface of an LNP or a nanoparticle. You may enrich about 2 or 3%, but the rest is going. Myeloid cells, spleen, and liver. So obviously, as the company's name suggests, Myeloid Therapeutics, we're actually quite happy that the myeloid cells are so good at taking up all of these lipid nanoparticles. And so we said, look, why don't we focus on using protein biology instead of LNP technology to drive expression selectively of our CAR? And this is shown schematically on the top left here. This is just a make-believe protein on the cell surface. It's made of 2 subunits, A and B. For this to be expressed on the surface, the 2 subunits come together in the ER, and then they get shuttled to the surface. What's really important here is if you just express subunit A without subunit B, or vice versa, you won't get any cell surface expression. And so what we did is we took half of the receptor. So you can see here, we've fused an scFv tumor recognition domain to it, and that's what we actually express as an RNA construct and deliver that directly in vivo. And what happens is you only get arming of cells that have both subunits and the ones that don't. So within the innate immune system, actually the immune system in general, there's a lot of multi-subunit receptors. We started with IgA. So the IgA receptor is Fc alpha. It's a very, very inflammatory receptor. There's actually quite a bit of data showing that high levels of this receptor within tumors is positively correlated with survival. But it's made of two subunits, CD89 here in the orange, and then the Fc common gamma chain here in the red. And so what we did is we took the CD89 component, we fused a tumor recognition domain, and that is what our CAR is, delivered as an RNA LNP complex. And the idea here is that myeloid cells which express the Fc common gamma chain will express the CAR on the surface, and cells that do not are unable to express a functional receptor. So when we started Myeloid Therapeutics, and even today, we definitely face the question of how do we know that the innate immune system is going to deliver great results in the clinic. And so what we did to try and solve that from the perspective of delivering data that's meaningful is we started working in models where CAR T cells are ineffective and so is checkpoint inhibitors. So this particular model here is a B16 melanoma model. We use it just as a model to look at activity. If you give CAR T cells at the same time as tumor seeding in this model, or checkpoints, they don't work at all. What we actually did is we waited for the tumor to be seeded, we waited for it to grow to about 75 to 100 millimeter cubed, and then we initiated treatment with the CAR that's targeting GP75. It's a protein on the surface of this particular cancer model. And as you can see on the right here, when we fish out the tumors, only the myeloid cells express the CAR. But what's really, really amazing, on top of the great clinical activity, is that when we look in the entire mouse, we look at the entire adaptive immune response, we find that within lymph nodes and the spleen, activation of antigen-presenting cells, dendritic cells. When we look in the tumor, we see accumulation of activated T cells, and we see a reduction in exhausted T cells within that compartment. And obviously, we're not developing it for curing mouse cancer, and lots of people have already done that. What we really want to do is build a product that can move the needle for patients. And we landed as our first target into TROP2. TROP2, if you've been following the ADC space, is something that's heating up as a target. It's a very widely expressed epithelial tumor target. But what you can see here, I apologize. What you can see here is overall responses of 35 to 45%. And so we actually feel that there's a significant opportunity for us. And so what we actually did, to mitigate risk but to push us faster into the clinic, is we embraced the tumor targeting domains of Trodelvy. So we took the CDRs from Trodelvy, we fused that to our CAR. That's what our construct is. We also did a deal with Acuitas. These are the folks that have been developing LNPs. For example, they're the folks that provided the lipid nanoparticles for the Pfizer BioNTech COVID vaccine. And so by using proven lipids and also a binding domain that we have good experience around, we're able to accelerate into the clinic. And we actually think we have significant mechanistic advantage over these ADCs based off the fact that this is not an immune suppressive therapy. ADCs tend to be immunosuppressive through their ability or side effects of killing myeloid cells, for example, and so on and so forth. So first and foremost, what we did is we actually went into a xenograft model. So on the left here, you kind of see the diagram. It's the anti-TROP2 scFv fused to the CD89. We encode that as an RNA, and then we went into a xenograft model. So this is really important here because we're not activating T cells. We're only activating myeloid cells. There's no T cells in this model. We don't provide any T cells. So all this shows is the direct anti-tumor activity of armed myeloid cells alone. And you can see that it's quite potent, and on the right-hand side, you can see we're getting a good 10% arming of the myeloid cell compartment within these particular animal studies. And so what we actually know from a mechanistic perspective now is the CAR actually functions through multiple different mechanisms. The primary mechanism is activation of the full immune response, but first and foremost, we know that an armed myeloid cell can actually kill tumor cells directly. It's a little bit of phagocytosis, but not really that much. Most of the direct killing is actually through TNF and other trail-like pathways to kill it. What's really interesting and what we're very excited by is the activation of NK cells, T cells, the accumulation of these cells within the tumor, and we actually even see activation of B cells, which you can actually detect through the production of tumor-specific antibody. So why are we excited by this? The whole idea here is the ability to get into the tumor, recognize the tumor, and then liberate neoantigens. And so we believe this technology really has shown, at least in the animal models, and we're in the clinic now, this ability. We obviously, on our path to the clinic, went through non-human primates. We've shown that we can get anywhere from 20 to 40% specific expression of the CAR in the circulating myeloid compartment. We don't see expression in any other immune cells within the non-human primates. So we're super excited by that. And so where are we? So we're dosing patients. We've dosed 2 patients today. We're on the dosing schedule is once every other week. And so it's a basket study looking predominantly, obviously, at safety and tolerability. But over the next 3 quarters, we'll continue to collect data, and we will move quickly based off the data set that we've been creating. So I'm going to stop there for a minute and just kind of shift to how we build transformational products at Myeloid. Might be a little cliche, but I really look to folks like Andy Grove, who was the founder of Intel, and I'm always walking around telling our scientists we need to make sure each product is 10 times better than the one before it. And so we really are building the company with that perspective, and I think if you look at starting with the cell therapy and now recapitulating that directly in vivo, that's a pretty transformational leap. And I think where we are right now for our next programs or our next products, we're on the same path. And I'm just going to show you very briefly two elements to some of the things that we're doing. The first thing is T cells still take up lipid nanoparticles, 3 to 5%. NK cells take them up too. So there is this push to try and drive for immune cell specific LNPs. I think that this is a very difficult thing to do. What we've been focusing on is just taking advantage of what LNPs already provide. And so we've actually moved into developing very specific CARs for different cell subsets. So what you can see here is a number of different CAR constructs across here. So CD89, CD16, if you've been following Affimed, this is a NKN myeloid-specific receptor. NKp44 is also. But the most important thing is you can see that they all need to find their binding buddy to be expressed on the surface. And so we actually took NKp44 and all of these and have shown that you can actually specifically activate or arm different immune subsets. And remembering that, unlike virus, RNA has a theoretical capability of 14 kb, right? So you're talking 3 to 4 times larger than any virus, for example. The other thing that we've been doing is using AI to assist with the development of our RNA constructs. On the left here, what you'll see here is just using AI to help design codon optimization with the idea of structure. We take structure very much into account. We believe that structure is actually what drives RNA expression and stability. And what you can see here, just by using AI to help increase the free energy, you can see that there's kind of almost like this bell-shaped curve of expression where we took a CAR that couldn't even be expressed. We just changed the codons to shift and create a few hairpins and so on and so forth. And you can see we're changing the expression profile, and we can get up to almost 8 to 10 times the level of expression by using this approach. The other really interesting thing is we've discovered a microRNA site specific to, in this particular instance, myeloid cells. And this microRNA binding site is unique in that it stabilizes mRNA. Often microRNA is used to stop expression. In this particular instance, when we introduce the microRNA into the non-coding region of the RNA construct, we can actually get expression of the RNA up to 120 plus hours. So we're seeing expression of our CARs up to 12 days. This is actually, I haven't actually seen any other RNA construct that's really out there, and this is this is a linear RNA. I saw Tom sitting there. And so we're very excited by this because what this really does is give us, therapeutic index. You saw the expression with the non-human primates, 20 to 40% of circulating myeloid cells. If we can then also, increase the duration and, level of expression, that's significant. And using linear RNA is really important for what we're doing at Myeloid because we also have been working on using RNA constructs to deliver genes. Imagine if on one RNA construct, for the cost of the COVID vaccine, you could do sickle cell engineering, you could do all of your genetic engineering, no viruses, nothing. And we've essentially been working towards that using line 1 retrotransposons. We're running out of time here, but I think what the most critical element to this to point out here is by taking these line 1 elements, and these are elements that are within our genome, they've been there over billions of years, we've actually been able to take them, we fuse a Cas element to it, and we actually, with site specificity, deliver genes directly into a safe harbor site within the genome. And so we're very excited by this technology because we think it's the natural progression for our company as we're developing capabilities within RNA delivery and understanding how cells take up RNA, how it's used. The ability to use the same technology to deliver genes, I think, is going to be very transformative, especially for those using viruses today. So just to summarize where we've come as a company and how we see the world, hopefully I've convinced you we have a versatile platform that's broadly applicable. We believe we're disruptive, obviously going straight into the clinic with CARs and RNA, and our ability to move this into NK and T cell programming over the future will show that. We really strongly focus on putting products into the clinic and learning from the clinical experience, and we're very fortunate to have investors who believe in that approach and supporting us through that progress. Our experience in non-human primates and now in humans is really helping us as we move forward. And as you can probably appreciate, we've obviously proven our ability to scale, GMP manufacturing, and working with partners like Acuitas and so on and so forth. We bring all of that to the table because it does take cities of people to move these products, as I mentioned to you, in 19 months from an animal experiment to the clinic. So where will we be this time next year? We should be on the other side of our dose escalation from our 302 program. Hopefully we'll be, GPC3 liver cancer study will be becoming initiated and we'll be accelerating our GeneWright technology into the clinic. So that is the end of the presentation.
Great. Thank you for that. Super interesting. I think we have time for maybe one or two questions. First, I'll open it to the audience if anyone has questions. Otherwise, I can jump in. So for your clinical program with TROP2 that you mentioned, so you talked about having, I think, data later on, I think 2024. Does the trial incorporate a biopsy? Kind of, what will you be looking for to signal that your program is working?
So we have a baseline and day 50 biopsy.
Mm-hmm. Got it.
So yeah, we'll be looking for that, yeah.
Mm-hmm. Got it. And so you'll be looking at things like, I think you showed in the preclinical data, things like activation of the immune system in the tumor and all of those kind of parameters.
Yeah, so we're using spatial RNA-seq, so we'll be looking at over 900 different genes and then also using the IHC to be able to layer on top of that. So we can look at myeloid-specific genes, T-cell-specific genes, and then also start to understand, which patients we think will respond and not respond, because obviously in a phase 1 study, you're kind of a little bit at will there with, luck, mm-hmm. right? Because that's, it's a very small patient population. Mm-hmm. So obviously the focus is on safety. We deliver this RNA every 2 weeks. And we've done actually weekly in non-human primates without any significant toxicity. So we believe that will be fine, and then we'll go from there.
And with that broader immune activation mechanism, do you expect there to be activity in tumors where TROP2 expression is heterogeneous?
Yeah, I mean, obviously that's the plan. Mm-hmm. We'll have to see it. I mean, we see epitope spreading in the animal models. Uh-huh. And so we hope to see that obviously in the human situation as well. Mm-hmm.
Got it. And then just in the last 45 seconds or so, you talked a little bit at the end about the retro-T and the kind of genetic engineering technology. How are you thinking about applications outside of oncology?
Yeah, I mean, the ability to deliver with site specificity a gene and all of your editing tools with one strand of RNA is completely transformative, I think, for all gene editing. And so we're actively looking and working with partners within the space, because obviously you think of the companies like CRISPR, Editas, so on and so forth, Intellia, this could really transform, the delivery of those technologies. So I think the sky's the limit for it. It's beyond the purview of Myeloid, and that's why we look for partners to help with that.
Great. So perfect. I think we're just about at the end of our session. So thank you again for the great presentation and discussion.
Thank you.